Hi, sorry I'm not with you guys today. I am actually in a meeting with the Department of Education for the U.S., including getting ready to meet the Secretary of Education, Arnie Duncan. So, sorry I'm not here, but I'm thinking about you, trying to make things better for you. I just want to do a real quick review of some important things that you need to get from each of these assignments before your test tomorrow. When we did this assignment about heroin versus morphine, we were actually really just looking at the functional groups and how this part of the molecule is very similar, but these two parts are different. And because this one is more polar, hydroxyl more polar, this will actually be able to, this one is actually held back from crossing the cell membrane, and this one is actually able to cross the cell membrane. So the shape actually matters more than the size does. We then wanted to kind of understand the importance of shape and then just kind of look at why plants might be producing this. Over here we were really looking at feedback and how the binding of one thing can actually influence all the other steps. And so if this negatively inhibits that. So making sure that you really understand what a receptor is, but mainly talking about feedback there. Next, we want to look at the pH inquiry. And of course, you can always pause the video as you're shuffling to get your sheets out, and then start it again. But the pH inquiry, mainly what we need to know here is what is pH a measure of? How do I measure it? Remember log and writing it in exponent form, and then looking at which one's more acidic, which one's more basic. What does it actually mean? Keeping in mind that I didn't put POH on here, but that pH and POH together actually equal 14, and that's where that 7 comes from. Being able to then analyze the scale and talk about how much more acidic, giving it a quantitative number. And then finally being able to kind of read a graph and look at an example of using a graph to kind of tell what's going to happen. Keep in mind that if carbon dioxide dissolves in water to become acidic, what does that mean about the number of hydrogen ions there are? So this assignment, we really want to know that property of water. There are lots of other properties of water, so make sure you go back and review your properties of water also. Next, we talked about carbon, and so you do want to take a second in your notes and look at what was going on with carbon. Why is carbon unusual? What are hydrocarbons? But then we went in to talk about what a carbohydrate is. And so we should be able to identify the basic structure of glucose, the monomer, and how I can put the monomers together in different forms based on what I need structurally, but also keeping in mind structure relates to function. So what is the difference between starch and cellulose? What is important about glycogen? What is important about chitin, about peptidoglycan? They have some things in common, but they're also very different. And then we wanted to use this article to help us understand the difference between using carbohydrates in plants. They are supportive as cellulose, and they store extra energy or food, sugars, as starch, versus humans who use them for energy, break them immediately down for energy, or store them as glycogen. And so we do kind of want to use that article to help explain that. Here we talked about being able to use carbohydrates to tag. So the shape of the carbohydrate is actually very repetitive. So it's not going to be great in terms of shape, but it's going to be easy to access. It's also an easy polymer to build. Using dehydration synthesis, taking out water, joining them together, we can actually build ones of all different sizes. And we can get a little difference based on whether they're a branching chain or a straight chain, alpha bonds or beta bonds like in starch and cellulose. And so, of course, we know this is actually a protein, and so the protein is really the better shape tag. But we can throw these carbohydrates on to make even more diversity. So that was our carbohydrate that we wanted to know about. Next, we had lipids. And And so keeping in mind that lipids are also made of C, H, and O, but that they're combined very differently. We've got a glycerol backbone, just a few oxygens, and then these fatty acid tails. And so what you needed to go through and do was identify that all lipids are C, H, and O, that the majority of lipids have a glycerol backbone and fatty acid tails. Going back to know what does saturated and unsaturated mean, these of course being saturated with hydrogens and flat and stacking and solid at room temperature found in animals. These having carbon-to-carbon double bonds. And so you really should be able to look at these diagrams and tell what's going on and how the structure ... relates to the function. So taking some time maybe tonight to go back and make sure you know how the structure relates to the function. Keeping in mind we have these steroids that are actually lipids. If they don't have the glycerol backbone, these are going to be quick signaling molecules. So instead of having to build a giant protein to be a signaling molecule, we can use these hormones to be signaling molecules. When we analyzed a lipid, we actually were looking at membrane fluidity, jumping ahead a little bit to talk about the cell membrane, which is made of phospholipids. And what does it mean if a phospholipid has a saturated tail or an unsaturated tail? What does it mean if the phospholipids are close together or spread apart? And how does cholesterol or other fatty acids actually play a role in membrane fluidity? And then looking at how lipids can actually be hormones, and that's good. They're mostly nonpolar. Lipids are mostly nonpolar, hydrophobic. So they're usually going to have to be carried by something in the blood. That's not good. But once they arrive at the cell, a lot of times they can actually pass or transverse the cell membrane very, very easily. This means they can go in and have an immediate effect. Here we're having an effect on protein synthesis and which proteins are being made. And so knowing that the disadvantage is that it's not going to travel well in blood. If it has to bind to a receptor, then it actually has to have what we call a secondary messenger that will help the signal propagate or continue. So keeping in mind the advantages and disadvantages of being a lipid really have to do with that whole layer, the nonpolar. Lipids are nonpolar. And so that can be good about crossing the cell membrane, but it can be bad about how it interacts with the rest of the cell and the external environment. If the cell is mostly water, sometimes we're actually going to need a little carrier to carry it. So keeping in mind all those hydrophobic properties. Next, we analyzed nucleic acids, and first we just defined them. So how can we identify all nucleic acids? They're all polymers. They're all made of repeating units of nucleotides, keeping in mind that a nucleotide has 3 parts: a pentose sugar, a phosphate, and a nitrogen base. So all nucleic acids are polymers or chains of nucleotides. But nucleic acids differ based on the type of nucleotide used. In DNA, deoxyribose is the sugar. In RNA, ribose is the sugar, and we're not seeing that sugar over here. In DNA, the nitrogen bonds are adenine bonding with thymine. Nitrogen bases are adenine, thymine, guanine, cytosine. So the nitrogen bases that are part of each nucleotide come in 4 forms in DNA. In RNA, they also come in 4 forms, but we've got uracil versus the thymine here. The other things you need to know are how many rings are in each of these. You need to know which ones are purines, A and G, the big ones, pyrimidines, C, T, and U, the nitrogen base, is a pyrimidine. Knowing that how many hydrogen bonds, A, T, we get out of school at 2. And then talking about the bonding patterns in which ones bond. We also want to talk about the functional difference, storing information versus actually producing the protein. And there, for our intents and purposes, we said there were 3 types of RNA. But Mandy and Sarah Bonin actually introduced interfering RNA to you. So there's much more to RNA than just the ones that we talked about. And so this is where base pairing comes into play. Both of them base pair. DNA base pairs to itself. RNA base pairs to DNA when mRNA is being made, if you remember. But mRNA up here in this picture also base pairs with tRNA. And so we want to kind of know what's the advantage of base pairing, what's the disadvantage of base pairing. Down here where we evaluated nucleic acids, we actually wanted to look at what is the advantage of DNA, of having two strands connected. What's the advantage of RNA, having just mostly single strands that sometimes fold on themselves? And so being able to talk about why DNA might be what we're using now and why we still need RNA. And so that is where we are evaluating nucleic acids and even looking back to maybe what did the first cells look like in that primordial soup, your A's, T's, G's, C's, U's, which ones were there. And then finally we've got analyzing proteins. And of course, this is where we spend a lot of time. So you should really be able to identify the 4 levels of folding. You should really be able to talk about how not all have all 4 levels of folding and how the interactions between these levels really create different shaped proteins. And again, how does shape relate to function? When we analyzed a protein, we wanted to compare what effect does a protein misfolding have directly? So what does it matter if a protein is misfolded? What does that matter? What's the local effect right around that protein? Keeping in mind, it might be an enzyme, it might not. And what are the systemic effects? What does this mean for the whole body? How could it be that one misfolded protein gives us a whole huge series of disease symptoms? And then finally, evaluating proteins and looking at how all kingdoms have hemoglobin. Even bacteria, why would they have hemoglobin if it's to transport blood? And knowing that it can even do more than transporting oxygen. So maybe we want to talk about what they have in common and what does this mean? So evaluate the statement that proteins perform specific functions in light of the statement above. So what does it matter that they perform specific functions? If I read this, what does it matter that they do specific jobs? Of course we want to know that it has to do with the shape, and it has to do with how those jobs might mean that the cells have a lot in common. And that's where the second part comes in. If I give you a series of amino acids, that's what this diagram is. We've got 1, 2, 3, 4, 5, 6, 7, 8 different hemoglobin protein amino acid sequences. So if I look at the hemoglobin in a human, in a soybean, and in this protist, this single cell organism, they all have hemoglobin, but how much do they have in common and how much are they different, and what does that tell you about the organism? Okay, one more thing that I want to talk about in your packet, and that's this quiz that you got back on Friday or today, Alberto. And keeping in mind that your test is not fill in the blank, but that this is kind of an okay way to start structuring your study guide, leaving yourself blanks that you know you need to go back and answer again and again. We don't really need to know about the linkages as much as being able to recognize the prefixes and suffixes of parts of the word. But you really, really need to know this part about the protein folding. What is caused by what? So, good luck on your test tomorrow. 47 multiple choice questions, 3 short answer. You can only answer them in a paragraph. And then 1 full essay. And I did go ahead and tell you that that'll be on enzymes. When you come in tomorrow, you know what is in your packet. Please have it in order and go ahead and staple it. Miss Woodall will collect them, and then we'll just leave them on a lab station. Good luck.